Hi guys, so we're starting the DNA unit. I'm gonna go ahead and hit concurrent slide, there we go. All right, so we're starting DNA. It kind of rolls right after blood into DNA. So just like if I already have a blood sample and they actually want to do the extra step to try to get DNA out of it, now here we are. So how do we get that DNA out of it? What is the processes? What are the lab techniques? What do they do? And what is it? So introduction, DNA or deoxyribonucleic, okay, deoxyribonucleic acid. There you go. Hard word to say, which is why we call it DNA instead of deoxyribonucleic acid, is a genetic material in our cells. So no two people except identical twins have the exact same DNA. So identical twins do have the same DNA, because remember what an identical twin actually means is they were the same egg, they were the same sperm joined together, but for whatever reason, that egg split into two. So it has the exact same genetic material, just split in half. If you are a fraternal, what's that? Fraternal? What's the other kind of twin? Okay, I don't know the word. The other type of twin that is not identical does not have the same DNA. So it's going to be two different eggs, two different sperms, two different things. And every person is going to have a slightly different DNA train. So DNA patterns from 4 sets of twins, which are identical here. So if you're seeing these DNA patterns and they should be identical, matching down the bottom. Okay. So DNA fingerprinting is also known as DNA profiling. It is a technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. So that's, if you've heard of DNA fingerprinting, it has nothing to do with your fingers, nothing to do at all. It just means that, so like a fingerprint is unique and individual to you, so is your DNA. So that's why they call it DNA fingerprinting. What a DNA fingerprint actually ends up looking like is what this man is holding here. So this is Sir Alec Jeffreys. We've talked about him before. He's the first person that invented DNA fingerprinting and used it in the first crime scene case in 1990. So this is what it looks like. It prints out, and if people have the exact same bands, the exact same thickness, and exact same placements, then they have the same DNA. So we're going to talk about how this process is made more and more, but first off, you have a blood sample, they extract the DNA out of the blood sample, and we will do a lab on how to extract DNA from something. It is different depending on the substance. We're going to pull it out of strawberries because that's really easy to do, but they pull it out of the blood first. Their enzymes are added to your DNA that cut it at specific points. Basically, it's like they have enzymes that recognize the same sequence in your DNA and make a cut there. DNA fragments are separated by their size. So depending on where the cut is, maybe this cut had, like this long of a strand, and this cut had this long of a strand, and this cut had this long of a strand. So they're going to be cut at different points. And the links of those cuts end up shaking out based on their size. So like the bigger the DNA piece is, the less far it can make it, because it's heavier, longer, it's not going to make it as far. And the smaller pieces make it towards the bottom. They eventually settle out here into these bands. And we'll talk more about that. It's high level concept, just already kind of introing it. So DNA fingerprinting has been used to help solve crimes by linking suspects to the crime and by helping identify bodies of victims. So if we find a body, we don't know who it is, you can use DNA to figure out who it is. Kind of example for how that could be done. Let's say we have a missing person, their body's too decomposed, they don't have dental records, we don't know who it is, but we look at our missing persons list in the area and we find a couple matches that seem to be the same sex, the same size, around the same relative age of our body that we have found. How we could try to figure it out is we could go to the relatives of that missing person, take their DNA samples and compare it to the DNA of our body and see if they match, if they could have came from the same family origin. So if we're looking at this little crime scene sample up here, you see this is the sample taken from the crime scene and it matches suspect 2. So again, this is circumstantial evidence. It doesn't mean just because someone's DNA is at the scene that they did the crime, but it does mean that they were there at the crime scene. In addition to a crime investigation, DNA fingerprinting can also be used to establish paternity. So I just gave you that instance of we have a missing body. Well, so how would paternity help us? Well, if I have a missing body and I don't have their DNA on record, they've never been to prison, so their DNA is not on record or anything, then I could take their parents, match them, and see if it matches our missing body. So that could be how it helps us in a crime scene. But also just like paternity. So like if you have, you're the father, like how do you figure that out? Or they do a DNA test. So if you're looking at these bands here, so this is our mom bands in blue, dad's bands in yellow. And when we're looking here, the children, sorry, I'm yawning. The children of this couple cannot have a band that doesn't match one of them. Like you get your DNA from your parents. So you cannot have a band that doesn't match them. It has to match one of them. So like in this sample, so she got this band from her mom. She got this band from her dad. She got this band from her dad. She got this band from mom. So that's their kid. Whereas I look at this one, she's matching the mom, but she has nothing from the dad, and she has bands that don't match either of them. So what that tells us is that he's not the dad. So DNA paternity testing can indicate that a man is highly likely to be the father without 99% accuracy, or that he's excluded. So like, nope, can't be because your DNA isn't in there. In addition to crime investigation, DNA fingerprinting can also be used too. So this is kind of like the list. So when we just talked about paternity, showing you how you match them, they have to match at least one of the parents. Can't have a band from like some unknown place, or that unknown place would be the other parent that you have. Identify victims of war on large scale disasters. So that's just a sad thing that is a reality. If you have a lot of victims of war, most of the time the bodies are all placed together in like mass graves. So later when family members come back to try to see if their family member died in the war or in the large scale disaster, like a hurricane, something like that, all they can do is take those bodies that have been found and, like I said, take the DNA from the family members and see if they find matches to those missing persons. If they do, then that's probably their family that they found that died in the disaster. Study biodiversity of species, so we can see how diverse our species are, how mixed match their DNA are, see how related by species are. So like if you heard that we do have the same, some of the same DNA as a banana, well, that's because they've checked it. We have some DNA that matches. Track genetically modified crops. So seeing if our genetics are being passed on the way we want in our modified crops that we're creating. Settle immigration disputes. So if you have people living in an area and they're disputing who's been there longer, you could look at old records, DNA type them to see which one do they match more so of who's been living in this area the longest. Most lab techniques used for DNA fingerprinting were not intended for those purposes, but instead were developed for the medical field. So a lot of what we're doing is trying to identify issues in the DNA strand to see if you have a medical diagnosis, diseases, things like that we can see. That's where a lot of this stuff started. That's how Sir Alec Jeffrey started, was he was not trying to solve crimes. He was just trying to find pieces that were incorrect in the DNA and then realize that he could create a DNA fingerprint that could also help in crime scenes. Small amounts of biological evidence left at crime scenes called trace evidence are the source of DNA needed for DNA fingerprinting. So some places that we do leave our DNA is saliva, blood, semen, skin, hair roots. Remember, if I just have my hair strand, there's no DNA in it. But if you rip your hair out and it has that root, that follicle attached, body tissue cells, so like obviously hunks of your body, and even urine can sometimes have some DNA in it. And here's other couple things. So bone, teeth, sometimes even feces and vomit. A lot of times that's because there's blood in it though. Okay, so for structure and function of DNA, DNA is, let me escape out of this for a second. Yeah, I think I want to do like 2 more slides. Okay. Just making sure. So structure and function of your DNA. So DNA is a nucleic acid. You might remember that for biology. For some of you, that was 4 years ago, though, so maybe not. But it's a nucleic acid found in your chromosomes. Okay, so you see these chromosomes here inside. So we have a cell. You have a nucleus. Remember, our red blood cells don't have a nucleus, so they don't have DNA in it. Only our white blood cells do. But any of your cells that have a nucleus have your chromosomes in it. There are 46 total. So 23 pairs of chromosomes, 46 total, and they're all in the nucleus of your cells. Exception is in your sperm and egg cells. We talked about this when we talked about the Punnett squares, how of like blood genes, so they get passed down because obviously if each sperm had all 46, then 46 would meet 46 in an egg, and then you'd have 90 something. Adding math there. Okay, so that'd be too many. We're only supposed to have 46. So the way that that works out in our like reproductive systems is sperms only have half. So they're going to have 23. The egg's going to have 23. When they come together to make the baby, now you have 46. And that, so they have the correct number of chromosomes. But every other part of your nucleus and cells has all 46. Just in your sperm or egg cells, depending on which one you have, there's only 23 in those. Each chromosome pair can be broken into smaller segments called genes. So, like this little hunk here is a gene. All a gene does is it controls a trait of an organism, and therefore they vary. Okay, so like if you have the brown eye gene, okay, tall gene, like those, all those, your blood type gene, all that is like just basically a section. So it's a smaller segment of a chromosome broken down in pieces. Here's a gene, it codes for something in your body. Your genetic code, DNA, has 4 different nitrogenous bases. So basically, this code, you can think of it kind of like a computer code, like codes of zeros and ones. It's a code of A, T, G's, and C's. And they are just nitrogen based little molecules that are paired together in a specific way. And when they are paired together in a certain order, they code your body for a certain gene. These bases form pairs according to the base pairing rule. So adenine always pairs with thymine, so AT, or TA, but they're always going to be paired together. Cytosine binds with guanine, always. Guanine goes with cytosine, cytosine goes with guanine. They're not going to go with anybody else. These pairs are considered to be complementary. So if I tell you that this side is an A, you know the other side is a T, because that's how they pair. A way to remember it, apples go in the tree, cars go in the garage. Okay, so C's with G's, T's with A's. Apples are in the trees, cars are in the garage. Now you can think of other acronyms, but I know that's one that I've been taught. It's one that sticks with me. Like I don't even have to think about that one. It always stays with me. So if I forget which one goes with which, I'm like, oh yeah, apples and trees. And if you can remember that one, then the other ones go together. So cars in the garage, cytosine, guanine. So if one strand of DNA has a genetic code ATC, TGC, what would its complementary strand be? So if A is always paired with T, then I know there's going to be a T paired there. If this is a T, well, an A is always paired with T, so this is going to be an A. And I have a C, well, G's are always paired with C, so this will be a G. Back to a T, T's always get an A, so there's going to be an A. G's always get a C, C's always get a G. So it look like that. So you would know what their pairing would look like. That'd be your little genetic code there. Okay, and that's where we're going to pause for today's notes.